So, hi everybody, I'm Matthieu Chourrout, based in London within Claireworks group here. And I will present the extremely brilliant brain, which is the Isotropic Microscopic Micrometric human brain dataset. So, well, you all know the brain atlases, which can be derived from microscopy, MRI, or even light chain microscopy. But with those, we are usually limited to a resolution of 0. Well, in most of the cases, maybe 0.5 millimeters. And so that's why we are trying, and also we have different constraints about the 3D integrity of the data. And this is why within our group, we are using virtual histology, which is 3D isotropic, high resolution, and designed for the whole organ. In order so first we digitalize the organ and then analyze it on the computer digitally. So for that, in my group we use phase contrast X-ray imaging, which is more sensitive than absorption compared to, for example, clinical CT scanners. So for example, with absorption contrast, you will get mainly the bones. But with phase contrast, you will be able to see most of the details within the sample. So for that, we use synchrotrons, and most specifically the European beamline, the European synchrotron and the beamline BM18 in Grenoble. So I won't go into the details of the synchrotron, but basically it's a big machine in order to do X-rays. And those X-rays are very intense with a huge dose, and they are highly coherent. So basically, in order to get the signal, the phase contrast, we just shift the detector away and use what's called free propagation. This way, depending on the distance, if we increase the distance, we will get good edges, and we will be sensitive to this phase contrast. So basically, in our group, we designed the hierarchical phase contrast tomography. Basically, we scan the sample at first a resolution of about 20 microns, maybe 25, and then we use it in order to screen the sample, and then we do another scan at a resolution which can be higher, up to 2 microns, for example. So, for example, we can scan different regions within the brain and then use these different scales in order to have good insight into the structure. So I will go briefly into this. And so, for example, we will have in blue we have the 6 micron resolution, and in yellow we have the 2 micron resolution. But then, in my case, I won't be talking actually about the 20 micron resolution. I won't be doing hierarchical phase contrast tomography here, but I will do instead 7.72 micron resolution scans. So this data set is called the extremely brilliant brain. This is a proof of concept for a high resolution whole brain scan. So basically, if I go through the methods, so it's ex vivo, so we get the brain postmortem, which is extracted by our collaborators. For example, in the case of this brain, the donor was male, 63 years old. The brain is supposed unaltered because the donor died of pancreatic cancer. Then we go through a process which is fixation and mounting, which also involves dehydration of the sample. And finally, we do the imaging. And this yields an image with an isotropic pixel size of 7.72 microns. The volume is about 20K by 20K by 20K, which yields a file size of about 13 terabytes. So I will now go through the volume. So yeah, as I said, it has a lot of slices, and the whole data is isotropic. And here we get maximum intensity projections, and we can see all the different details within the brain. Oh yeah, and sorry, and this was done in 24 hours. So then I will detail some workflows. So first we can study the anatomical features. We can also study the blood vessels because the data is label-free. And then we can also study the white matter tracks because the dehydration will increase the contrast within the white matter. So first we worked with Timo's team actually in order to integrate the data set into the BigBrain space. And the two data sets are quite complementary. Indeed, we can get fine parcellation, a common coordinate system, and even the cell types when we do histology with the BigBrain dataset. And from the extremely brilliant brain, we get labor-free data, which is isotropic, and we also get a fast throughput of 20 hours for a scan. Now I will detail another type of analysis workflow. So basically, the idea is that you can browse the data through a web browser, and then you can decide of an array which you'll be studying. So then you can download to a small workstation, which doesn't have to be a Buffy cluster, for example. You can do the segmentation, the analysis, and then bring back the data within the online tool. So I did this with cortical vessels, which can be studied which can be tracked down and studied through a different length. So for example, I was able to track over 5 millimeters, one of the vessels within the cortical layers. And finally, in order to study white matter, we can do what's called structure tensor, which can be related to diffusion studies. And so, for example, if I zoom in, we are able to see the white matter tracks here, and this will lead in the future to the analysis of the white matter tracks within the whole brain. So in conclusion, the dataset is soon to be publicly released. We presented how to use it with dedicated tools. And so in the future work, we want to use different tools. For example, I will get in touch with Lindsay about using different tools for parcellation. We are glad to benchmark all your tools if you want to run a collaboration with us. And also, our goal is now to develop core studies in order to tackle some biomedical challenges about some diseases. So I would like to thank you all, and I'm happy to answer any questions and also to browse the data. Thank you.